Hello students, welcome back to a new video of Sunanda's tutorial. In this video, we will discuss about Speroplast. What is Speroplast? Speroplast is a cell that lack the complete cell wall. The cell where the cell wall is completely or partially removed. So basically this is the meaning of Speroplast. So this Speroplast is present in your UG botany syllabus, in major syllabus as well as minor syllabus according to new education policy. Okay, let's start. So it is a microbial cell from which the cell wall has been almost completely removed by the action of penicillin. By using penicillin, we can remove or we can inhibit the formation of cell wall. Then we can use lysozyme. According to some definition, the term is used to describe gram-negative bacteria. Actually, cell wall-less gram-negative bacteria. It is appropriate in case of gram-negative bacteria. The term speroplast stems from the fact that after the microbe cell wall is digested, membrane tension causes the cell to acquire a characteristic spherical shape. So after the degradation of cell wall, the overall cell take a characteristic shape. Speroplasts are osmotically fragile, okay, and will lyse if transferred to a hypotonic solution. So in a hypotonic solution, if we place our cell in a hypotonic solution, then water will enter inside the cell by the process of endosmosis. So after endosmosis, the cell swell and finally it will break in case of speroplast because they lack tough or thick or hard cell wall. When used to describe gram-negative bacteria, the term speroplast refer to the cells from which peptidoglycan component but not the outer membrane component of the cell wall has been removed. So actually in case of gram-positive bacteria, they have cell membrane and outer to the cell membrane thick peptidoglycan layer is present. But in case of gram-negative, we have cell membrane, then peptidoglycan, then lipopolysaccharide layer. So in case of gram-negative, simply to form a speroplast, we have to remove that peptidoglycan component. Okay. So again, what is L form bacteria then? So sometimes we got confused between L form and speroplast. What is the difference between L form and speroplast? L form bacteria actually cell wall deficient bacteria, or this is a variety of bacteria those lack the cell wall. They are named after Lister Institute in London where they were discovered. Okay, but there are some variety of Bacillus genus, Bacillus subtilis species. Those lack the cell wall and called as L form of Bacillus subtilis. Okay, two types of L form are distinguished. One is unstable L form that are capable of dividing but can revert to the original morphology, that means with cell wall, and stable L form that are unable to revert back to original bacteria. So speroplasts are a type of L form bacteria that can divide and also revert back to their original form. Speroplasts are created when the cell wall of a microbe is completely removed, causing the cell to take a spherical shape. Then how speroplast can be formed? One is antibiotic induced speroplast, because when we apply antibiotic, they inhibit the synthesis of cell wall, and finally cell wall less form of microbial cell can be produced. Some examples are chloramphenicol, oxytetracycline. Okay, so there are some peptidoglycan synthesis inhibitors such as phosphomycin, vancomycin, mionomycin, beta-lactam antibiotics, antibiotics that can inhibit the biochemical pathway directly upstream of peptidoglycan synthesis, induce Speroplast 2. There are some antibiotics which are inhibitor of protein synthesis, inhibitor of folic acid synthesis, and this causes gram-negative bacteria to form Speroplast. So by using different types of antibiotics, we can induce Speroplast. Next, enzyme-induced Speroplast. There are some enzyme like lysozyme that can cause the Speroplast in gram-negative bacteria. Okay, next is what are the uses and applications we can use for antibiotic discovery. Okay, so inhibitor of cell wall synthesis caused growing the bacteria to form Speroplast. This screen enabled the discovery of phosphomycin, cefamycin. Okay, so in this way we can screen whether the antibiotic can inhibit the bacterial cell wall synthesis or not. So we can discover antibiotics, then cell lysis. So yeast cells normally protected by cell wall, which makes extraction of cellular proteins too difficult. But enzymatic degradation of cell wall with gymolase creates Speroplast of yeast cell, renders the cells vulnerable to easy lysis with detergent or rapid osmolar pressure changes. So after enzymatic digestion, we can extract the protein from yeast cell. Next, what is transfection? A bacterial Speroplast with suitable recombinant DNA, which is genetically modified DNA, inserted into them and can be used to transfect animal cell. So we can easily insert our modified, engineered or recombinant DNA. That means DNA with our suitable gene or gene of interest. So DNA piece with our gene of interest, those can express our one suitable character can be inserted inside the bacteria and can be used to transfect animal cell. After infecting the animal cell with that bacterial cell, we can produce proteins inside the animal cell. Okay, our desired proteins or any character. Then Speroplast with recombinant DNA are introduced into media containing animal cell and then fused by polyethylene glycol. And with this method, nearly 100% of animal cells take up the foreign DNA. That means the animal cells can be made competent to uptake the foreign DNA. So in this way, this Speroplast have different functions. So basically it is all about Speroplast. I hope you really enjoyed this session and get benefit from this in future. And if you liked it, please hit the like button, share with your other friends, and please subscribe to my channel Sunanda's Tutorial. Thank you.